Hello, and thank you for your interest in my talk. My name is Alex Price, and I'm a postdoc in the lab of Matthew Weitzman at the University of Pennsylvania and the Children's Hospital of Philadelphia. Today, I'm very excited to tell you our story about how a DNA virus can prevent sensing via double-stranded RNA by promoting efficient splicing of its own viral transcripts. In general, double-stranded RNA is considered a hallmark of viral infection. Among these, RNA viruses, whether of a positive sense genome or an antisense genome, can, during the process of replicating their RNA genomes, must make a full-length genome copy of double-stranded RNA during this process. DNA viruses, however, are a slightly different story. While these DNA genomes never have to go through a double-stranded RNA intermediate, they do often make transcripts coming from both the top strand and bottom strand of the DNA. These symmetrical transcripts can combine via base pairing and therefore make, again, long stretches of double-stranded RNA that can be sensed by many different factors of the innate immune system. However, this is slightly controversial within the DNA virus field. Among cellular double-stranded RNA sensors, there are three major categories. These RIG-I-like receptors, or RLRs, that ultimately signal through interferon and the production of interferon-stimulated genes to combat infection. Second, there is this protein kinase R pathway, which upon sensing double-stranded RNA can auto-activate its kinase domain, blocking the translation through phosphorylating of eukaryotic initiation factors. Third, there is a pathway called oligoannyl synthetases coupled with RNase L. This OAS RNase L pathway, upon sensing double-stranded RNA, forms the secondary molecule 2' 5' oligo A that activates a latent RNase inside the cells, cleaving both viral and cellular RNAs, leading to apoptosis and ultimately cell death to block viral infection. Now, DNA viruses have been long characterized to be in an evolutionary arms race with basically all of these pathways. Shown here are a number of DNA viruses from diverse clades that now all encode different inhibitors of essentially every single pathway of this double-stranded RNA sensing and response to viral infection. In particular, our lab studies adenovirus, a DNA virus known to encode both a transcription factor that blocks the interferon signaling of RLRs, as well as a small structural RNA called the virus-associated or VA RNA that is well characterized to inhibit PKR by essentially binding its active site and sponging it from being able to detect actual double-stranded RNA. However, the actual direct evidence that these DNA viruses are producing double-stranded RNA inside living cells is lacking. We decided to explore this more with our adenovirus infections. To do this, we wanted to be able to look at double-stranded RNA formation in situ during infection. We used an antibody raised against the A-form helix of double-stranded RNAs, one of which here is called J2 and was able to easily sense transfected double-stranded RNAs in the form of poly-IC. Surprisingly, however, when we looked during infection with human adenovirus, what we saw was no cells that showed any amount of double-stranded RNA, both with this wild-type infection as well as with our mutant that lacks the PKR sensor, the PKR inhibitory molecule VA RNA. This really led to this question of what conditions would actually cause adenovirus to make double-stranded RNA, or else why make this PKR inhibitor? When we look at the transcripts that come off of adenovirus, we see that adenovirus is a relatively simple virus with about a 36 kilobase double-stranded piece of DNA as its genome. Its transcripts are moved into two different classes, these early transcripts, which are thought to be primarily distinct units and ultimately involved in antiviral responses and genome replication. However, upon entry into the late phase, there is now this single promoter, this major late promoter, that through the process of alternative splicing and alternative polyadenylation makes many different viral late transcripts. Now, when we look at this genome, we can see that there is many places in which double-stranded RNA might be predicted to form. In this core region, though, what I'm highlighting is that it would actually be exons of these late strands base pairing to introns of the opposing strand. Furthermore, read-through events caused by poly-A cleavage read-through could lead to transcripts where, again, we're having an unprocessed transcript base pairing to a processed transcript from the opposing strand. This really highlights that it might be RNA processing itself that is controlling the formation of double-stranded RNA in this infection. To ask this question, we wanted to know is we wanted to look at specific viral mutants that are known to control viral RNA processing. Of these, there's this really interesting factor made up of two viral proteins that together hijack a host colon-based ubiquitin ligase. This now virus-directed ubiquitin ligase redirects this ubiquitin post-translational modifications to degrade antiviral factors that would otherwise attack the viral genome. However, none of this explained the RNA processing until our lab recently did a proteomics approach where we saw multiple cellular RNA binding proteins were now non-degradatively modified by ubiquitin by the same virus-directed ubiquitin ligase. This explained a phenomenon in the field for decades where it was known that in viruses lacking either of these factors of this virus-directed ubiquitin ligase, the virus would have very poor processing, splicing efficiency of its transcripts, and these transcripts would often be retained inside the nucleus. So we want to know, using these mutants, can we ask whether poor RNA processing of the virus can lead directly to double-stranded RNA formation? And this appears to be exactly what we saw. Using 3 different mutants of of the different pathways of this virus-directed ubiquitin ligase, we now see abundant double-stranded RNA forming inside the nucleus of these infected cells, and only forming in the cells that have reached the late stage when these opposing transcripts would be present. However, this really led to more questions than we answered, one of which, the biggest one being, where is this nuclear double-stranded RNA sensed? When we go back to the effectors that I've highlighted before in these existing double-stranded RNA-responsive pathways, we see that all 3 of the sensing units are predominantly residing inside the cytoplasm. So does our nuclear double-stranded RNA, is it activating or even detected by these things? And if so, how? We first focused on the PKR pathway and looked at autophosphorylation of the kinase as well as downstream factors. And what we saw is that while wild-type infection is not activating the PKR pathway, mutants that lack this ubiquitin ligase and now produce double-stranded RNA robustly activate PKR during infection. So this double-stranded RNA is capable of being sensed. Furthermore, when we looked at PKR's localization, we saw that in uninfected cells or wild-type infected cells, PKR retained its cytoplasmic localization. However, when we looked inside mutant cells that produce double-stranded RNA, we now saw PKR relocalizing into the nucleus and co-localizing only in the cells that were producing double-stranded RNA. Secondly, we looked at the RNase L pathway. The best way to look at activity of this latent RNase is just by looking at the integrity of ribosomal RNAs, shown here by a bioanalyzer gel. What we see is that during wild-type infection, we do not see any evidence of RNase L activity whatsoever. However, upon the double-stranded RNA producing mutant, we now see these cleavage products informing that RNase L is active, and this is dependent upon RNase L being present in these cells. Furthermore, we were able to characterize the upstream oligoadenylate synthetases and find out that, again, it was the cytoplasmic form, OAS3, that was absolutely critical for activation of RNase L upon sensing this nuclear viral double-stranded RNA. Now, though we did really need to know what was the source of these double-stranded RNA molecules, as DNA virus infection has been shown before to upregulate cellular endogenous elements that could form double-stranded RNA. To ask this question directly, we were able to use our J2 double-stranded RNA antibody to perform an RNA immunoprecipitation. What we saw is that our positive controls of cellular mitochondrial RNAs or invariant repeat alloelements were able to be robustly pulled down, yet they showed no difference between the wild-type mutant virus infection versus the mutant virus that produces double-stranded RNA. When we looked at viral transcripts, however, we saw that while our mutant viruses were producing many of these transcripts were robustly enriched during infection, when we looked at splice-specific qPCR, highlighted here in these hashed bars, it was specifically the unspliced or poorly processed form of these viral transcripts that were most enriched by the double-stranded RNA pull-down. Now we wanted to really ask, how does this ubiquitin ligase itself actually regulate RNA processing? This is really summarizing a huge amount of work spearheaded by an excellent graduate student that's no longer in our lab. But what we have found was that this ubiquitin ligase primarily modifies 2 different cellular RNA binding proteins, HNRMPC and its homolog Raleigh. When this ubiquitination event happens, these factors can no longer bind viral transcripts, and this allows for their efficient processing. In contrast, in the ubiquitin ligase mutant viruses, Raleigh and HRMPC can directly associate with viral transcripts and block their splicing. Importantly, we were able to show that a simple removal of these antiviral RBPs was able to restore viral RNA processing efficiency. This led us to ask, would such a thing impact double-stranded RNA formation? So here again, when we do a double knockdown of Raleigh and HRMPC, we see that this restores the protein expression during mutant virus infection, and again, restores the splicing efficiency of these late transcripts. Now, very excitingly, what we saw is that upon knockdown of these two RBPs, we saw either no double-stranded RNA formation or substantially reduced double-stranded RNA formation in these mutant virus infections, really implying that increasing the splicing efficiency was sufficient to decrease double-stranded RNA production. However, these mutant viruses, as well as these highly abundant cellular factors, have many different functions in the cell. And to really ask the very specific question about splicing efficiency, we wanted to take a different approach, in which we know that splicing is actually directly catalyzed by RNA-RNA interactions targeting 5' and 3' splice sites. By blocking these with antisense morpholino oligos, we're able to specifically impair the splicing of certain transcripts while sparing others. We designed antisense morpholinos that target viral late transcripts alone. So by transfecting these oligos, we do not affect the splicing of an off-target viral gene, and yet robustly impair the splicing of on-target viral late transcripts. Now, when we look for double-stranded RNA, we see that for the first time ever in wild-type infection, by directly blocking splicing efficiency, we now see double-stranded RNA inside the nucleus of these infected cells, really providing mechanistic evidence that it is splicing efficiency itself that is preventing the formation of double-stranded RNA during these infections. To summarize, we set out to ask the question, what happens when adenovirus RNA processing is defective? We learned that while the wild-type virus alternatively splices and processes its own transcripts so efficiently that there is no detectable double-stranded RNA during infection. In contrast, when mutant viruses can no longer modulate this process, we now see accumulation of abundant double-stranded RNA inside the nucleus of these infected cells. Ultimately, this nuclear double-stranded RNA is activating both cytoplasmic antiviral pathways inside the nucleus. With that, I would just like to thank my lab, Matthew Weitzman, who was my mentor in this whole process, really let me take this project on, as well as Susan Weiss at Penn, who helped us with all the double-stranded RNA antibodies, as well as the OAS RNase L pathway. I'd also like to thank my funding sources, including a very recent NIAID Pathway to Independence K99 award. If you or anyone you know is interested in taking on a new researcher that would love to ask questions about RNA processing and double-stranded RNA sensing inside the nucleus, please contact me. You can do so at either my email address here or on Twitter, shown here as well. Other than that, here's a model of my talk, and I would love for anyone in the future to reach out to me with whatever questions you might have. Thank you very much.